So Year 11s, today we're going to be looking at active transport. But before we get there, I wanted to do a quick recap of diffusion and osmosis. So there are three factors I wanted to cover again with diffusion and osmosis. The first is that diffusion occurs down a concentration gradient of the solute. So if we're looking at the diffusion of sodium, we're looking at it going down its concentration gradient. Water occurs down its osmotic gradient. And the way that we determine the osmotic gradient is slightly different, and you'll need to refer back to your slides for that. They're both passive processes, so they both don't require any energy, but because they don't require any energy, both of these stop when equilibrium is reached. Now, a lot of chemicals are required in concentrations much higher than they would be if they were only achieved at their equilibrium, if they were only moved across at their equilibrium. So this brings us to this need for something else, a type of active transport, a transport where there is energy that's involved. So we are able to, with active transport, we're able to push against the concentration gradient. We're able to move past that equilibrium and start to actually move molecules against their concentration gradient. Now obviously this is going to require energy. If we use the example of a concentration gradient being like a hill, it's very easy for a car to roll down a hill. You can turn the engine off and you can stop the engine running, you can take your foot off the accelerator and the car will move down the hill. But with active transport, we're moving up the hill. And so we need to put the foot in the accelerator, we need to expend some energy. So this is an active process. It requires energy. So we're going to go into this in a little bit more detail. Now active transport doesn't have to occur only up the concentration gradient. It doesn't have to go against the concentration gradient, but it is able to. And so this is a really important thing. The part that drives active transport, there are two processes that drive active transport. So active transport occurs via protein pumps. And this is for small molecules or small chemicals. And then there is also the movement of large molecules, which occurs via endocytosis and exocytosis. And we'll go into these two in more detail. We'll start with the protein pumps though. So with protein pumps, what we're going to be looking at is a considerable amount of energy, a considerable amount of substances that are going to be moving across here. So the protein pumps, they look at small molecules. We'll just use our... So we're going to cover this with just a diagram. So we have our cell membrane. Here is our protein pump. Now on the outside here, we have our solute, whatever it might be, and on the inside we have the same solute. Now I've drawn that there is a concentration gradient here, so if we quickly just do that up, in the cytoplasm versus the ECF, if this is our concentration, then we have a comparatively low to high. So these molecules are going to naturally want to move into the cytoplasm, but they're impermeable to the membrane. So they're not able to cross the membrane in any other way. So this is our membrane here. They're not able to enter in, but the cell needs them. So what it's going to do is it's going to use this protein pump. The protein pump, in order to work, and we'll just use our arrow once again, so it needs energy, and that energy allows it to change shape. So that we go from this where it's closed, where there's no entry into the cell, to having entry possible. So this energy actually changes the shape of the protein pump, and it allows us to pump in more molecules. And in this case, we're going to do so until, and this is what protein pumps do, they will do that until the energy is exhausted, until they have run out of energy. So this is a constant process. Protein pumps allow for very rapid exchange. They work extremely efficiently, or extremely quickly rather. But this energy intensive process does mean that there needs to be some controls. So the body or the cell actually does control the use of these protein pumps by restricting the amount of energy that's available to them. But that's the basic process there. So we can create an imbalance using protein pumps. The other situation here is when we're moving large molecules. So we're moving macromolecules, or we're moving large amounts of molecules at once. So we'll have our new heading. Moving macromolecules, and first we're going to look at endocytosis. Now, if we look at the individual words here, endo means into, and cytosis is just cells. So it's into cells. Endocytosis is bringing substances, bringing these macromolecules into cells. So the process is actually relatively simple. We have our cell membrane once again. And this time our cell membrane, instead of having a protein pump, it's got an integral protein. So it's got a protein that goes all the way through, and that integral protein has a sugar attached to it, which is able to detect a particular substance. So let's say that this substance is a fat molecule, a large fat molecule. And this particular cell wants that fat molecule. Maybe it's a liver cell, or maybe it's just a cell that's wanting to store more energy. So it encounters this, and then it creates its own shape change. The cell membrane actually begins to change shape around that molecule. And eventually, the cell membrane reforms, and what we'll end up with is a vesicle with that fat inside. So here we have our vesicle. And then that vesicle could go on, and it can be broken down, the fat can be digested and broken down, and it can work rather well. That's for a solid particle. Now the same is true for a liquid particle, we just change the name. So pinocytosis, and the pino is just drinking, and then the cytosis once again is cells. So we're talking about pinocytosis, we're talking about the movement of liquid particles. So we're talking about cell drinking. Again, these terms, if you're aware of them, they're not particularly difficult. But this is our process. We have a particle, we identify that particle using a receptor of some description, it's almost always a protein. That then creates a changing in shape, and this changing in shape is called invagination. So we have this change in shape, this invagination of the cell membrane, and then we eventually form our vesicle, and the vesicle will go on to do whatever it is that it needs to do. The second process, pinocytosis and endocytosis are related to each other because they're both going inside the cell. The other process, though, is exocytosis. Now, exocytosis, again, we have this cell term, but then exo means exit. So what are we doing here? We're getting rid of something from the cell. So if we have our diagram, we have our whole cell membrane again. But this time, we've got our vesicle, and let's say this time our vesicle contains something else. Maybe it contains a neurotransmitter. So this is a protein. These are lots of hundreds of little proteins that actually need to get out of the cell. They need to go and do some work in the cell. And so the cell receives a signal to say that it needs to release these. So the vesicle itself attaches to the cell membrane. And when it attaches, the cell membrane actually changes shape. To fuse with the vesicle, and then the vesicle, then the contents of the vesicle are ejected into the extracellular fluid. So this is a really important processes here. We're moving either large volumes or just physically large substances into, or in the case of exocytosis, out of the cell. Remember going back to our cell requirements. We had these three requirements: energy requirements, material requirements, and waste requirements, or excreting requirements, excretory requirements. And you can see now how these three processes, diffusion, osmosis, and active transport, how they're involved in this. This is a really, really critical thing that we need to understand. So with exocytosis and endocytosis, we have this ability to move large molecules. With protein pumps, we can move small molecules, but very quickly. With facilitated diffusion, where we're using protein channels, we're able to move small molecules very quickly, but only down their concentration gradient. With all of these molecules, particularly for endocytosis and exocytosis, we have this need for recycling at times. So perhaps that molecule that was endocytosed, that was taken into the cell, was a damaged cell particle. Instead of that fat molecule that we draw, maybe it was a bit of organelle, maybe it was a bit of cell. And that's going to have valuable resources, valuable nutrients. And so the body then has this need to get those resources. And there's a special structure that it uses within the cell, stored in vesicles, and these are called lysosomes. These vesicles are called lysosomes. And those lysosomes contain enzymes that are going to break down these substances. So these are, once again, the body doesn't want to waste anything. It spent time making these cells. It spent time making these organelles. It spent time making these proteins. So it needs to be able to get them back. So it has these vesicles, these lysosomes that contain digestive enzymes to do so. Anything that's useful will be recycled, and anything that's waste will be packaged and sent out into the cell, either through exocytosis or through protein pumps or channels. We've got a few worksheets there, worksheet 8, and we also have some chapter 1.5 questions. That brings us to the end of chapter 1.5.